Hi, my name is Bedbach, and here's my project proposal, which is a rough plan for an experiment, which includes CRISPR removing the beta secretase gene in order to potentially cure Alzheimer's disease. Alzheimer's disease is a neurodegenerative disease that leads to the progressive loss of synapses and neurons in the brain, which leads to deficits in learning and memory, behavioral and cognitive decline, and brain atrophy, as shown in both figures in humans. There are two different types of AD, the familial side and the sporadic side. Familial is caused by genetics and it makes up 3 to 5% of all cases. Sporadic is caused by unknown factors. It makes up 95 to 97% of all cases. In this experiment, we will be attempting to solve both cases. AD pathology is caused by three known factors. We have the amyloid beta plaques, which are formed by the APP protein being incorrectly cut by the beta secretase enzyme in order to form A beta proteins that congregate to form these plaques that inhibit neural function. Then you have the tangles, the intracellular neurofibrillar tangles, which are structures inside the neurons that unravel to inhibit neural function. Then you have chronic neuroinflammation, which is caused by the immune cells of the brain, the microglia, reacting to the A beta plaques. And these immune cells then release chemical factors that cause inflammation in the brain and kill off neurons. Here are two diagrams of A beta plaques form. Then here is a GIF of that same thing. They are congregating to form a plaque. And then here's my hypothesis. I will be using CRISPR to remove the specific genetic sequence that codes for the beta secretase gene or base 1 in an animal model to examine any side effects of that process and to see if the risk of neurodegeneration is reduced. We will be using CRISPR as a technique to delete the beta secretase gene. It is made up of two different things, Cas9 and gRNA. Cas9 is the protein that cuts the DNA, and the gRNA, or the guide RNA, tells the Cas9 where to cut. Here's a rough sketch of my project. We will be taking the genetic sequence for base 1, completely deleting it, and hopefully plaques no longer form in the brain. You'll be using the guinea pig because the guinea pig can develop A beta plaques naturally and have proteins that are very similar to humans, surprisingly. We will be taking a pair of guinea pigs. We will be taking their zygote, and then we will be editing that zygote, removing the beta secretase gene. And then we will grow out the zygote, take the offspring of that genetically altered guinea pig, and we will run multiple tests on that guinea pig. We will then run the open field test to test for any abnormal behavior to see if any side effects are caused by removing this gene. Then we will be taking them to the Barnes maze to see if Alzheimer's disease still affects them. Then we will use genotyping to confirm that the pups do not have the base 1 gene. Then we will sacrifice the guinea pigs to use their brains in order to find any sort of A beta plaques in their brains. And if they do not have any A beta plaques in their brains, then that means we were successful in removing the base 1 gene and eliminating all A beta plaques. Our expected results is that we will be investigating the role of the beta secretase, which is caused by the side effects. And then we will also hopefully get rid of all A beta plaques and effectively removing two pathologies and for AD. Unfortunately, I do not have the correct resources for this project, nor do I have enough funds. This is impossible to complete without a proper lab. And if I were to be taken on by a lab and if the experimentation goes smoothly, then we will be moving on to primates and then hopefully to humans in order to successfully cure AD. Here are my references, and I think you have a great day.